The topic of this lecture is neurological disorders. Although the brain is our most protected organ, it's encased in the bony skull, it is cushioned by fluid, and it's surrounded by 3 protective layers of covering. There are still many diseases and pathologies that can hurt the brain and severely damage and disrupt its functioning. So a neurological disorder is any disease or dysfunction of the central or peripheral nervous system that causes damage. There are over 600 known disorders currently, and people are affected by these worldwide. The World Health Organization gives numbers such as 50 million people worldwide are affected by epilepsy, 62 million people are affected by cerebrovascular disease like stroke, 326 million people suffer from migraine, and 24 million are affected by Alzheimer's disease and other dementias across the world, and that number is only expected to increase exponentially. The World Health Organization advises that undergraduates learn about these diseases and disorders because they estimate that 20 to 30% of the population will develop one or more of these disorders. There are multiple causes for neurological disorders, and I will talk about a few. The first one is genetic, and these are diseases caused by faulty genes. Developmental diseases are problems with the way that the nervous system develops. Degenerative diseases include Parkinson's and Alzheimer's, where nerve cells are damaged or die. Cerebrovascular diseases are diseases of the blood vessels that supply the brain, such as stroke. Traumatic brain injury and injury to the spinal cord are another source of neurological disorders. Seizure disorders, which include epilepsy, cancer, such as cancerous tumors, and finally, infectious diseases are another source of brain disorders. The first one that I will talk about is genetic brain disorders, and I'll talk about inherited metabolic disorders first. There are at least 100 different metabolic disorders that can affect the development of the brain. So normal functioning of cells requires these intricate interactions between lots of biochemical systems in the brain. So a metabolic disorder usually results in an inability to synthesize an enzyme or protein that's needed in the brain for development. And this lack of the enzyme can often cause brain damage or impair development if it's not identified quickly. If it's identified quickly, then it can be treated to prevent serious brain damage. The most common metabolic disorder known is hard to pronounce. It's phenylketonuria. This disorder results in the absence of an enzyme that is needed to convert the amino acid phenylalanine to tyrosine, and excessive amounts of phenylalanine interfere with the myelinization of neurons in the central nervous system. And since much of the myelinization of the brain occurs after birth, the brain fails to myelinate and develop normally, and this results in severe retardation. So diagnosing PKU immediately after birth is really important so that the infant's brain is never exposed to high levels of phenylalanine. And as a result, many governments have passed laws that mandate tests for metabolic disorders like PKU to prevent cases of mental retardation and brain damage. So I have 2 more examples of inherited metabolic disorders, and the first one is pyridoxine or vitamin B6 dependency. And this is a very rare disorder caused by a recessive gene that interferes with the enzyme that is needed to use vitamin B6. So infants present with seizures within 2 hours of birth due to pyroxidine deficiency because of the lack of that enzyme, although there are some kids that don't experience symptoms until they're 2 or 3 years old. The treatment for this one is very large doses of vitamin B6, because if the infants aren't able to get that vitamin, then it will result in damage to the white matter in the brain. Galactosemia is another inherited metabolic disorder that results in a missing enzyme needed to metabolize galactose, which is a milk sugar. And if untreated, this can also cause damage to the axons or the white matter and to the cerebellum in the brain. This is very different from lactose intolerance, however, so don't confuse the two. And lactose intolerance is just insufficient production of the enzyme that breaks down lactose. And that only results in digestive problems, not brain damage. One of the most disabling brain disorders that has strong genetic links is schizophrenia. Between 1/3 and 1/2 of all homeless adults have schizophrenia, and 50% of those who have been diagnosed with schizophrenia have not received any treatment. The National Institute of Mental Health has classified the symptoms of schizophrenia into 3 categories. The first category is the positive symptoms, and these are psychotic behaviors that are not seen in normal people, and they include unusual thoughts, behaviors, sensory perceptions such as hallucinations, where the individual hears voices and thinks those voices are actually talking to them, which can be quite terrifying. Often a person with schizophrenia can seem quite normal until he or she talks, and then you're aware of the speech impairment. And in the words that they use, you become aware of how out of touch they are with reality. Sometimes they have disordered movement, either uncontrolled, jerky movements, or they may appear to be frozen or catatonic. The most common positive symptom experienced by schizophrenics is hearing voices. And these are called positive symptoms because they are added on. And these symptoms usually respond to drug treatment. A second category of symptoms is the negative symptoms, and these are the absence of normal behaviors. So thoughts and behaviors and emotions that normal people usually have, these individuals don't. And that includes lack of emotion or flat affect, monosyllabic or monotone speech, lack of interest in anything, and the inability to feel pleasure, lack of motivation, and also attentional impairment. Another symptom is neglect of basic personal hygiene. One of the problems with negative symptoms is that they are sometimes more difficult to identify because they resemble and can be mistaken for depression. So the last category of symptoms is called cognitive symptoms, and those include problems with the executive functions. So focused attention, working memory, the concept of time, the ability of the person to plan and organize, thoughts and behavior. Those are all dysfunctional. And this is very disabling because it affects that person's ability to function on a daily basis. So here's a diagram of the brain areas thought to be involved in schizophrenia. The first area, the basal ganglia, is thought to be involved in the movement dysfunction I mentioned. The basal ganglia, as you know, plays a role in movement, and the schizophrenic can display those jerky movements, or the opposite can appear to be frozen or catatonic. The frontal lobes are related to the difficulties that schizophrenics experience in planning their thoughts and their behaviors. And cognitive symptoms are also thought to be a result of dysfunction in the frontal lobes. The limbic system, which is involved in emotional expression and the experience of emotion, is also thought to be dysfunctional in schizophrenia and is exemplified by their lack of ability to feel pleasure and their lack of interest, and also the flat affect that they often experience. The auditory system is thought to be involved because of the experience of hallucinations when schizophrenics hear voices that they think are actually speaking to them, and also in the difficulties that they have sometimes in speech. The occipital lobes are hypothesized to play a role in the difficulties that schizophrenics have in identifying complex visual images and also in reading emotion on other people's faces. The hippocampus has been shown in schizophrenics to be atrophied and to have shrunk for individuals with schizophrenia. So one of the first proposed explanations for schizophrenia was the dopamine hypothesis, and that's the idea that dopamine in excess is what caused the psychotic symptoms in schizophrenia. So in response to that hypothesis, schizophrenics were prescribed medicines that blocked dopamine, and that was found to be successful for some people with schizophrenia, but not all of them. And more recent findings have shown that glutamate and its NMDA receptor are probably more important in the experience of schizophrenia. Specifically, glutamate metabolism, so too much glutamate, has been associated with changes in several areas of the brain that are related to schizophrenic behavior, including atrophy of the hippocampus that I just mentioned, and that atrophy has been shown to happen in the CA1 area of the hippocampus. What's interesting is that 50% of the people who have schizophrenia really don't have any awareness that they have the disease. And it's not because they're in denial about being ill, but It's thought to be because the areas of the brain that function to process this kind of self-awareness, parts of the frontal and the parietal and temporal lobes, have been damaged, potentially by glutamate in this disease. This is an image of 28-year-old identical twins, one with schizophrenia and the other one without. The one with schizophrenia is the one on the right. The healthy twin is the one on the left. So this image demonstrates two things. One, that schizophrenia results in measurable structural differences in the brain. You can see dark spots on the image on the right side. Those are enlarged lateral ventricles. So it's apparent that the cortical volume for the schizophrenic has been reduced. There's less brain tissue there, and that's characteristic of schizophrenia. So reduced cerebral volume and ventricular enlargement are things that go along with schizophrenia. And also, this image shows that schizophrenia is thought to be polygenic. Polygenic means that a trait or an attribute is the product of several genes together. But since these two individuals have the identical genome, it's more likely then that schizophrenia is epigenetic. And those genetic variations in combination with environmental factors like traumatic brain injury or early traumatic experiences or stress produce schizophrenia, because other factors may play a role in the expression of those genes that may have made a difference between these two twins, resulting in schizophrenia for one and relative health for the other. Attention deficit hyperactivity disorder is another brain disorder that's thought to have a strong genetic role. The estimated heritability of ADHD is high, and it ranges from 75 to 91%, which means that 75 to 91% of variation in ADHD behavior is linked to genetic factors. So several genes have been related to ADHD, but none have been definitively related as being the cause of ADHD. So ADHD in children resembles the behavior that one would expect with frontal lobe damage, and that includes the inability to maintain focused attention on a task or great difficulty maintaining that attention, difficulty concentrating, difficulty remaining still, and hyperactivity. And most children exhibit these characteristic behaviors at one time or another, but ADHD kids are different because they display these symptoms so often that they interfere with that child's ability to learn and also that child's ability to have social relationships. It's estimated that between 4 and 5% of all grade school children have ADHD, and boys are diagnosed about 10 times as often as girls. But this may be due to a failure to diagnose the disorder because it often presents differently in girls and boys. Girls often appear to be less attentive, whereas boys tend to have the hyperactive behaviors. Adults with ADHD show cognitive impairments and lower occupational attainment than would be expected by their education. And so the symptoms of ADHD in both kids and adults resemble those that you would expect with frontal lobe damage, specifically distractibility, forgetfulness, impulsivity, poor planning, and hyperactivity. Since the frontal lobes are also involved in short-term memory, dysfunction in this area in ADHD also disrupts the ability to remember to pay attention to important things, to monitor the effects of your actions, and to plan what to do next, because all of those things rely on the frontal lobes and working or short-term memory. So some have suggested that ADHD is related to deficiencies in dopamine functioning in the frontal lobes, and that those deficiencies are what produce the inattentive behavior. So drugs that increase dopamine activity have been prescribed to treat ADHD. And ironically, these drugs are stimulants. For example, one of the common drugs that's prescribed for ADHD is methylphenidate, which has the brand name of Ritalin. And that's a drug that inhibits the reuptake of dopamine. So it increases dopamine activity by inhibiting the reuptake of dopamine in the synapse. So it's there longer and it can hit that receptor more often to produce more functioning. So in this way, stimulants work a little bit like SSRIs work for serotonin in depression and anxiety. SSRIs work by preventing the reuptake of serotonin, whereas methylphenidate and Ritalin inhibit the uptake of dopamine. And making more dopamine available in the frontal lobes allows people with ADHD to function better. It improves those executive functions. Amphetamine is another drug that has been shown to increase dopamine activity and is also sometimes prescribed for ADHD. So the frontal lobe is thought to play an important role in attention-related behavior, but other brain structures are also thought to be important in ADHD. So multiple brain imaging studies have shown that there are abnormalities for people with ADHD, not only in the frontal lobes, but also in the dorsolateral prefrontal cortex, the ventrolateral prefrontal cortex, the cingulate cortex, the parietal cortex, the striatum, which includes the putamen and the caudate nucleus, and the cerebellum. All of these structures are key in cognitive and attentional functioning. But most researchers who study ADHD don't believe that it is any one of those structures by itself. ADHD is a result of abnormalities in functioning of the network of structures. And functional imaging studies lend support to this hypothesis. Another disorder with genetic links is autism. Autism tends to run in families, and twin studies have shown that there is a strong genetic influence for this disorder, although the number of genes and the specific genes involved are still a mystery. And it's currently thought that autism probably has an epigenetic cause as well. Autism and the autism spectrum disorders have been more common recently, and currently 1 in 88 people are likely to have an autism spectrum disorder. The classification of autistic disorders was recently changed with the publication of the DSM-5. But within this classification of autism spectrum disorder, autism is the most severe among a number of less severe autism spectrum disorders. The rates of autism have risen considerably in the last 20 years, especially in California, but it is really unknown right now whether this represents a true rise in the prevalence of autism or just better education and diagnosis by clinicians. Autism includes a spectrum of disorders, like I mentioned, but all of them include difficulties in certain areas: social interaction, verbal and nonverbal communication, and unusual responses to sensory experiences, such as repetitive behaviors. Autism is usually apparent by age 3, and after that age, it's not thought to further increase in severity. 50% of those who are diagnosed with autism, the most severe form of the disorder, are also thought to have mental retardation. By the way, recent studies have failed to find evidence that autism is in any way related to childhood immunization. And in fact, the investigator who originally claimed to have found evidence linking autism and immunization was found guilty of dishonesty for falsifying the data in that paper, and he retracted the article. An interesting aspect about the autistic brain is that, on average, it's slightly smaller at birth, but by the time the child is 3 years old, it's 10% larger on average than other children. Following this early growth spurt, the growth of the autistic brain slows down, and then by adolescence, it's only 1 to 2% larger than those without autism or more neurotypical children. 2 of the most common theories about the autistic brain involve the wiring of the brain. It's been suggested that the wiring of those with autism is different than the wiring of kids without, also called neurotypical brains. One of the hypotheses is that autistic brains are less well connected than normal brains, and those fewer connections between brain structures lead to difficulties that autistic people experience socially. So some studies have confirmed this underconnectivity with brain imaging evidence that show patterns of communication between different parts of the brain are not as synchronized as in neurotypical brains. And so it's been suggested that it's the fiber tracts or the axons that connect information from one structure in the brain to another that are the cause of autism. However, more recent theories have hypothesized that it's overconnectivity of structures in the brain that leads to autism. In fact, they call this overconnectivity hyperconnectivity, and it's hyperconnectivity that leads to social impairment in autism. Studies have also found evidence of greater connectivity in the brain, especially in the areas of visual processing. And this is consistent with recent findings of greater sensitivity of people with autism to auditory and visual input. They're more sensitive to sensory input, and they're faster at responding to sensory input. Other studies have found that the brains of people with autism are less active in parts of the dorsomedial prefrontal cortex. And in fact, one recent study examined treatment using TMS, which is transcranial magnetic stimulation, and found that stimulating that area of the brain in autistic children increased the activity in that part of the brain and significantly improved social skills. Other lines of research have focused on mirror neurons and reaction of people with autism to faces. So one hypothesis is that people with autism have dysfunction in their mirror neurons, or maybe a deficiency of mirror neurons. And mirror neurons are neurons in the brain that are thought to be involved in our experience of empathy because they actually fire when we see somebody else doing something. Mirror neurons fire when we see other people engage in behaviors, and they're thought to be partly responsible for our ability to feel empathy because the firing of our mirror neurons allows us to vicariously experience what another person is experiencing at the same time. Mirror neurons are also thought to be the reason why we yawn when we see someone else do it, otherwise known as contagious yawning. It's related to empathy. People laugh when they see other people laugh, and they're sad when others are sad, and that's the same thing with yawning. Our mirror neurons fire when we see the other person experiencing that feeling of being tired, and it causes us to feel sleepy and initiate a yawn. So the idea is that people with autism either do not have or have dysfunctional mirror neurons, and that explains the difficulty they have in understanding others' emotions and in social interactions. So one study hypothesized that people with autism would be unlikely to yawn after seeing another person yawning. And their study found exactly that. Watching yawns triggered more than twice as many yawns in neurotypical children than in the autistic ones, providing support for that idea. Another line of research focused on faces, because research has shown that the fusiform face area is less responsive in those with autism than those who are more neurotypical when looking at the pictures of faces. So this led some to speculate that people with autism are less interested in looking at faces and engaging in social interactions. However, a recent study showed that although autism is associated with less activity in the face processing parts of the brain when people with autism are shown faces, and that part of the brain that processes faces is called the fusiform gyrus, when people with autism looked at faces, another part of the brain showed much greater activity, and that was the insula. And this part of the brain is active when we feel pain and disgust. And that wasn't the case for neurotypical people looking at faces. So the authors speculated that one of the reasons people with autism hesitate to look in the faces of other people is because it actually is aversive to them. Here's an example of a brain disorder that people with the disorder probably don't think that they have a disorder. And this one is also thought to be highly genetic. The disorder I'm talking about is synesthesia. Synesthesia occurs when there is a crossing of the senses. So you experience a stimulus from one sensory modality like sound, and that triggers an experience in another sensory modality not typically related to it. For example, a musical note will evoke a color, or letters or words will be associated with a color or a smell or a sound. About 4% of humans experience this. So synesthesia is a harmless brain disorder, and it usually is a result of genes, but some people have reported experiencing it as a result of a stroke or a head injury. One thing that has been found is that synesthesia seems to be related to having strong artistic or musical ability. Another source of brain disorders is developmental effects that can be linked to diseases that are experienced by mothers when they're pregnant, or by exposure to toxic chemicals such as drugs and alcohol or environmental substances. These have all been shown to have a developmental effect on the brain. For example, if a woman contracts German measles, rubella, early in her pregnancy, the toxic chemicals that are released by the virus interfere with the chemical signals that control normal development of the brain. Most women who receive good health care are immunized for rubella, and that prevents them from contracting this disease during pregnancy. So this is not typically an issue in the United States. In addition to toxins that are produced by viruses, some drugs can also adversely affect fetal development, including alcohol. Mental retardation can be caused by the ingestion of alcohol during pregnancy, and the fetus is especially sensitive to the effects of alcohol during the 3rd to 4th week of gestation. So babies born to alcoholic women are typically smaller than average, develop more slowly, and have a condition called fetal alcohol syndrome. This image shows some of the common features of the faces of children with fetal alcohol syndrome. And there are not only effects on the face, there are neural effects as well. It's been found that alcohol disrupts the normal brain development by interfering with something called a neural adhesion protein. And this protein helps to guide the growth of neurons when the brain is developing. This disruption of this brain development is thought to produce these distinct facial features as well. So there are common facial features of children who have fetal alcohol syndrome, including a low, flat nasal bridge, smaller eyes, a shorter nose, the absence of the philtrum, and that's the groove on the upper lip, a thin upper lip, an underdeveloped jaw, and a smaller head. Now, a woman doesn't need to be an alcoholic to impair the development like this, because some believe that this syndrome can be produced by a single alcoholic binge during a critical period of fetal development. Here's an image of a normal mouse fetus on the left and one whose mother was fed alcohol during pregnancy on the right. So you can see that the alcohol produces similar abnormalities in the offspring of both species. The mouse on the right has facial malformations that are very similar to human malformations, including a smaller nose, a longer upper lip with a deficient philtrum. You can see that the lip on the alcohol-exposed mouse doesn't have that traditional groove, and smaller eyes with a narrow forehead. The mother of this mouse was exposed to alcohol during a time that's equivalent to the 3rd week of human pregnancy. And the effects of alcohol in this fetus were apparent within 24 hours of exposure. Of course, the facial features are relatively unimportant because much more serious are the abnormalities in the development of the brain. So children with fetal alcohol syndrome are born with many other issues other than the facial malformations, including difficulties with executive functions. Children with fetal alcohol syndrome have difficulty with focusing attention, planning, with understanding the effects of their actions. They often have behavioral problems including hyperactivity, lack of imagination or curiosity, social withdrawal, and anxiety. In addition to the facial malformations, other effects of fetal alcohol syndrome are a failure to thrive, developmental delay, organ dysfunction, even epilepsy. Prenatal alcohol exposure even appears to have direct effects on neuroplasticity. The offspring of female rats that are given moderate amounts of alcohol during pregnancy showed smaller amounts of long-term potentiation. And children with fetal alcohol syndrome also show learning difficulties, poor memory, less ability to understand concepts such as time and money, poor language comprehension, poor problem solving. So it appears that this finding generalizes to humans as well. The fact is that fetal alcohol syndrome is the #1 cause of preventable birth defects. So now that we recognize the dangers of fetal alcohol syndrome, pregnant women are advised to abstain from alcohol and other drugs that are not prescribed during their pregnancy while their bodies are engaged in the task of developing another human being, because these are the effects.